Okay. Hello. Good morning, folks, and welcome to our second day of class. I don't know if your home is any like my home, but I have some snow outside the window right now, so hopefully you'll stay safe today. Okay. We're going to get started intently with Chapter 1 content this morning. If any of you continue to have some technology issues with the course in terms of Wiley Plus or anything like that, I'd ask you to please hang on after class today, and we'll go through some of that individually to solve any problems you might have. Okay. So before we get started, are there any general questions you have at this time from last class that you'd like to ask? Okay, so gang, generally speaking, during a typical lecture, I will ask you to please always have your digital textbook open because through the lecture, I might refer to pictures and images in the textbook. And so I want you to have the ability to look at those images, bring them up on your computer, or if you have the physical textbook, to open that textbook up and have it so you can access it, so I can ask you questions about it, okay? So please, before you, even before you log in for every lecture, just make it a habit of opening up your digital textbook on Wiley Plus, okay? All right, so we'll get started at this time. I'm going to share my screen with you. And typically what I will do is bring up a Microsoft Word document that you see in front of you. I will keep open some video windows on the right side of my screen, and I will also have a chat window open on the bottom right. So if during our conversation, during my coverage of the content, if you have questions, please feel free to type into the chat window. And as I said, if there really is something urgent and I'm not addressing your concern, please speak up, unmute your microphone, and just interrupt me, okay? All right, so we're going to get started here. Chapter 1 seems like a good place to start, right? Introduction to the human body. Let's begin with some quick basic definitions that seem appropriate for our course. Okay, so the first is we should define what anatomy is, okay? What is anatomy? Anatomy is the science of body structures, okay? We're studying the structures of the body and the relationships between them. How do the muscles interact with the bones? How do the organs interact with each other via the bloodstream, via hormones, the electrical signals from the brain and spinal cord? You know, what is the relationship of these anatomical structures? Okay, we will, you know, in the process of this semester, particularly in lab, we will delve quite heavily in to the science of body structures. That's what lab is going to focus on. So that's half of your course. The other half of your course is physiology. So let's define physiology. Physiology is the science of body function, or an attempt to explain those body functions. How in the world does your kidney manufacture urine? How in the world does your liver absorb glucose? What does it do with the glucose? You know, how in the world does your brain generate electrical signals that communicate to far distant regions of the body, including down to your big toe? How exactly does that work? Well, that is under the domain of physiology. Okay, so anatomy is the science of the structures of the body, and physiology is the science of how those structures actually function. Okay, All right, so that's what we're going to be wrestling with all semester long. And as you pass into AMP2, you will continue that journey for basically an entire college year worth of content. So the first thing I'm going to talk to you about out of your textbook really is figure 1.1, figure 1.1. So I'm going to ask you a question, and sometimes I'll do this to sort of poke a sleeping dog. In other words, I want to be sure you are paying attention. Okay, so I'm going to ask you this in the chat window. And excuse me a moment. Okay, here we go. What is the title of figure 1.1 in your textbook? What is the title of figure 1.1? Please type that into the chat window for me. Okay. All right. Thank you, everybody that has responded and is currently in the process of typing in a response. I deeply appreciate that. You know, at least while we are remote, this is how a typical lecture will go, in that, you know, I want to be sure you're really looking at something that I need you to look at. So I'll ask you a question that requires you to look at it, so I know that you're actually looking at it. Okay. So thanks everybody for participating. We need to talk about structural organization of the human body. And so to do this, I'm going to bring up a website. I'm going to bring up a website, and I will actually put a link for the website in your notes. So the website I'm going to show you is the Genetics Science Learning Center, which is a learning center at the University of Utah. Okay, so let me copy this web link and I will put it in your notes. Okay, the Genetics Science Learning Center at the University of Utah. Okay, and there you go. You now have a hyperlink in your class notes to this website that I'm showing you. Okay, and so I will let you explore some of the website's features on your own. Okay, but the point here is that this is a supplementary learning resource for you. There's topics on genetics, topics on human health, of course, topics on neuroscience, topics on cell biology. Okay, so lots of things here you might want to play with, and you might find some interesting things here. What I'm going to do at this time is go into the cell biology tab. I'm going to go to Amazing Cells. Amazing Cells. I'm going to click on that, and there's a whole bunch of learning modules within this web space. And what I want to talk about and show you is this one here, cell size and scale. Cell size and scale. Okay, so I'm going to click on that and open it up. And what we're going to do is walk through some things in this interactive display on the University of Utah's website. You know, if I asked you right now, which is smaller, a baseball or a car? Well, obviously you would know the answer to that. I mean, the average baseball is considerably smaller than the average car. So with those kind of everyday questions and everyday topics, you already know the answer. It's disgustingly easy what the answer is. But when we start talking about atoms and molecules and other structures we might find inside cells, that intuition starts to break down, or at least if you're not used to thinking about the topic, it can be a bit uncomfortable. Okay, so we're going to walk through a couple things here, and then I'll document a few ideas in your notes, and we will move forward from there. So, okay, so the smallest piece of matter that's talked about in this interactive display is an atom. And in this case, we're seeing a carbon atom. Okay, so we're starting very, very small. You could, of course, ask, well, are there things smaller than atoms? And I think you know there are, right? Because you probably had physics in high school. So you know that atoms are made of subatomic particles, protons, electrons, neutrons, and even those things have smaller entities called quarks. But for our purposes in anatomy and physiology, thinking about your body, starting with the atom is a convenient and reasonable starting place. So here we have a carbon atom that's 340 ppm. So I'm going to ask you in the chat window a question. What does PM stand for in this display? What does it stand for? Okay. All right. And so, if somebody beats you to the punch in answering the question, please don't feel like you can't continue to answer. I want you to answer because I want you to be able to look at each other's answers and see if there's a consistency there or to see if there's disagreement. So you are absolutely right. A PM stands for a picometer. And you can, let me ask you this. How long is 1 picometer? Okay, and so, as you look at the website, hint, 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 as you look at the website, hint, 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 how long is a picometer? Okay, so you see that a picometer is 1 trillionth of a meter. A picometer is 1 trillionth of a meter. Okay, 1 picometer is 1 trillionth of a meter. And so what are we saying here? A carbon atom is 340 picometers wide. A carbon atom is 340 picometers wide. So a carbon atom is 340 trillionths of a meter wide. 340 trillionths of a meter wide. Now, don't worry too much. I'm not going to beat you up with heavy math in this course. There will be some math topics that we have to talk about. But, the point of it is that you start thinking from a scientific perspective, and science uses numbers, it uses mathematics. So let me ask you this. Here we go. How many inches fit into 1 meter? How many inches fit into 1 meter? Do you know? Do you remember from previous coursework you've done? How many inches fit into 1 meter? All right. All right. So as you ponder that, or perhaps as you Google that, I'm just going to keep rolling here. The answer is 39. So thanks for those that got it on the chat window. 39 inches fit into 1 meter. So let's think about this. If you had a meter stick between your hands, I bet you most of you have a yardstick at the house, right? A yardstick is 36 inches. So a meter stick is 3 more inches. So it's 39 inches. So if you had a meter stick between your hands and you were to divide that distance into 1 trillion evenly divided segments, and you counted up 340 of those segments within the meter stick, that would be the diameter across a carbon atom. 1 carbon atom is 340 picometers wide. Okay. All right. We're going to start to get a little bit bigger now. We're going to start to get a little bit bigger. Okay. So next we get to some things that we would more appropriately call molecules. Take note here. The water molecule is 275 picometers wide. Wow, what does that suggest to you? Doesn't that suggest to you that a water molecule is actually has a smaller diameter than a carbon atom? Okay, that might be a bit of a surprise to you. Okay, but think about it. Water is made of 2 atoms of hydrogen, 1 atom of oxygen, and they're held so closely together by their chemical bonds that the overall distance across the group of the atoms is smaller than the distance across a gigantic carbon atom. Okay. Now, that being said, the basic relationship and idea that we would ask you to take away from this is that, with exceptions, such as a water molecule being smaller than a carbon atom, with exceptions like that, generally speaking, atoms are smaller than molecules. Generally speaking, atoms are smaller than molecules. So we have methionine, which is an amino acid. So let me ask you a question in the chat window. Here it comes. What type of biological macromolecule contains amino acids? So in the computer right now, you see methionine, which is a type of amino acid, and it's giving you the dimensions there, 1100 by 700 picometers. So what type of molecule would contain an amino acid? Folks, I don't expect that you know, but I'm probing your knowledge right now to see if you do know, okay? So Brendan, you answer muscle. That's not wrong. It's just not quite specific enough. Okay. All right. So Jared has the right answer. It's protein, right? An amino acid is a building block of protein. Okay. All right. Here down here in the bottom right, we have adenine, which is a type of nucleotide. So let me ask you this. What type of organic macromolecule will contain nucleotides? What type of organic macromolecule will contain nucleotides? Okay, so adenine is a nucleotide. You can see its physical dimensions on the screen, 1300 by 760. Okay, good. So a nucleotide is a building block for nucleic acids. So what are the 2 main types of nucleic acids that exist inside cells? What are the 2 main types that exist inside cells? Okay. All right, very good, Jack. All right, it is DNA and RNA. Gang, if you're sitting at home and you're saying, I have no idea what the answers to these questions are, you know, should I already know the answers to these questions? No, not necessarily. So don't feel bad if you can't answer them right now. By the end of chapter 2, you will be able to answer these, okay? All right, so let's keep going. So we have carbon atoms, which are pretty much super, super tiny. And then we get larger objects, which we call molecules. Methionine is a type of molecule. Adenine is a type of molecule. We're going to put this into your notes, okay? We're going to do that. All right, we're getting some larger structures here now. Okay, so first off, let me mention glucose. Glucose is 900 picometers in diameter. Okay. Hold on a sec. I got a question coming your way. If I can spell with a darn. What is the name of the energy producing process that breaks down glucose? What's it called? Okay, so Jared, ATP is the name of the biochemical energy that's made by breaking down glucose. But what is the process of breaking down glucose called? Okay, so Lauren, you mentioned digestion, and Lauren, you're not incorrect. You're not narrow enough in your answer. Okay, so I'm all right. So take a look at Alana's answer. Alana's saying glycolysis, and Alana wins the prize for this question. Glycolysis is the name of the biochemical process used by cells to break down glucose. Okay, and typically that breakdown of glucose is shunted into another related process known as cellular respiration, which predominantly takes place in the mitochondrion of your cells to make your ATP. Again, I don't expect you to know it now. By the end of chapters 2 and 3, you will know this. Okay. All right. So we started with the carbon atom, which is super small, and we're getting a little bit larger with some molecules. Okay. So we have phospholipids, which you may or may not know, form the plasma membrane of cells, which is sort of the outer border. And you can see that a plasma membrane, you can see a phospholipid is 0.9 times 3.4 nm. So in the chat window, what does nm stand for? Good, it's a nanometer. It's a nanometer. Okay. So let's see here. I'm going to ask a specific student a specific question. If I call on you, please unmute your microphone and say hi to us. Okay. Naomi Myers. Naomi, please say hi to us.
Hi.
Good morning, Naomi. How are you?
I'm good. How are you?
Okay. So Naomi, here's my question for you. Is 1 nanometer larger or smaller than 1 picometer?
Larger.
You win the prize. Thanks, Naomi. You did great. Okay. So, what is it we're trying to talk about right now? We're trying to talk about the organizational size of objects that are inside your body. We started with the smallest things, which are atoms. Typically, the best unit to measure atoms is picometer, trillionths of a meter. Now we're getting progressively larger. We're getting to molecules that it would be more appropriate to measure them on a slightly different scale using a slightly different unit called a nanometer. A nanometer is 1 billionth of a meter. Okay. All right, let's get a little bit larger here. Okay, we have antibodies, which, on average, they're about 12 nanometers long, So let me ask a question in the chat window. What type of macromolecule is an antibody? What type of macromolecule is an antibody? Okay. I mean, everybody's worried about their antibody titers right now for COVID, right? I mean, if you've gotten vaccinated, everybody's worried about whether they have sufficient antibodies to protect themselves. Okay. All right. So, let me, Jack has posed a response, but let me get a feeling for how you're doing out there in the cyberverse, because I can't see you. I can't see your face. Here we go. Do you understand the wording of my last question? Give an honest answer. Do you understand the wording of my last question? Okay. So if you understand the wording of my last question, and only a couple people responded, that must mean you understand the question, but you just don't know the answer. And that's actually not a bad place to be. That's not a bad place to be. Okay, So as it turns out, Jack, you are right. Brendan, you're right. An antibody is a type of protein. Antibodies are made of protein. You're absolutely right. Okay. So antibodies are going to be larger than phospholipids, which are going to be larger than methionine, which is going to be larger than a carbon atom. Okay. Transfer RNA molecule, 7 nanometers long. Okay, 7 nanometers long. Question in the chat window. What do tRNA molecules do for a cell? What do they do for a cell? Anybody know? Well, okay, let me tell you. Transfer RNA molecules bind to amino acids. They bind to amino acids, and they deliver the amino acids to a machine called a ribosome, whose function it is to manufacture proteins. So tRNA is kind of like the Federal Express delivery guy delivering amino acids to ribosomes. Okay. All right. So let's continue. So, I'll spend some time poking you a little bit with questions, and then there are other times I'm just going to go faster so we cover content, okay? So, all right, so tRNAs are 7 nanometers. Hemoglobin, 6.5 nanometers wide. Hemoglobin is a compound that transports oxygen and sometimes carbon dioxide throughout your body, okay? It's made of two things. It's made of something called a heme pigment and a globin protein. And you'll learn all about it when you talk about blood. As it turns out, the topic of blood is going to be covered in AMP2. All right, we're getting a little bit bigger now. Okay, now look, here's the ribosome. We just said tRNA brings amino acids to the ribosome. You can see the ribosome is gigantic compared to tRNA. It is gigantic compared to hemoglobin. Ribosomes are the machines that make the protein, and they are much larger than a carbon atom, okay? Next, we have a hepatitis virus. Take a look. A hepatitis virus is 45 nanometers wide. A rhinovirus, which is one cause of common cold symptoms, is 30 nanometers wide. Okay. Here we have HIV, the cause of AIDS, the human immunodeficiency virus. We have influenza virus, which causes the flu. By the way, coronavirus, which we're struggling with the pandemic right now, is on the order of about 100 to 120 nanometers wide, so it's a pretty large virus. We have bacterial viruses. Yes, folks, bacteria get infected with viruses too, and that does alter their cellular physiology. We have coated vesicles, which are structures that enter or exit cells in the bloodstream, 90 nanometers. We have the measles virus there, 220 nanometers wide. And then we get to some other structures like a bacterial cell, E. coli. Take a look. E. coli is 3 by 0.6 It's not really a U, it's a Greek letter called mu. It stands for micrometer. A micrometer is 1/1,000,000th of a meter. So a nanometer is larger than a picometer. A micrometer is larger than a nanometer. Okay. Lysosomes or organelles, there are compartments inside cells that are involved in recycling worn out material. Mitochondria is the cell's power plant. You might have heard of this in other courses. This is where cell respiration really takes place, and you make most of your ATP when oxygen is available. Okay. Question in the chat window. do you notice that the physical dimensions of E. coli bacteria and the mitochondrion are very similar? Do you see that? That they're pretty much equivalent in size? I mean, there's, they are a little bit different, but they're very similar. Do you notice that? Folks, I want to tell you something that you're going to learn in microbiology whenever you end up taking it. Okay. And Brendan, I see your question. I'll answer that in just a second. Gang, mitochondria, scientists believe, and when I say believe, I don't mean we just believe it just to be fanciful. I mean, there is evidence to suggest. Scientists believe, because of evidence which suggests, that mitochondria are actually primitive bacteria that live inside our cells. Let me say it again. Mitochondria are primitive bacteria that live inside our cells. So that opens up a whole new way of thinking about evolution on the planet and how our cells came to be what they are. Mitochondria are primitive bacteria that live inside our cells. Okay? So it's what I'm trying to say here is it is no coincidence that the physical size of E. coli and mitochondria are very similar. So Brendan, you asked the question about bacteria and mitochondria. Does one go into the other? Not really, Brendan, because how could my left hand go into my right hand when they're the same size is what I'm saying. Okay. All right. Other questions? All right, let's get a little bit larger. Okay, so we're getting to some things like baker's yeast, right? Baker's yeast, maybe you use it at home to make some bread. I know some people still do make bread, and it's very tasty when it comes right out of the oven. 3 by 4 micrometers. Here we have a red blood cell, which is 8 micrometers long on average. Okay, so that's the typical size of the blood cells running through your body at this time. Let me ask a question here. All right. How many hemoglobin molecules are typically found inside one red blood cell? You already know, because we had it on the screen a little bit ago. There's hemoglobin, right? 6.5 nanometers. Hemoglobin. I'm asking you, how many of them are found inside a typical red blood cell? Okay. So Brendan, I see you qualified your response and you talked about iron, so I'll let that one go. And Jared, I appreciate the response, but you are very much too low on that. The answer is 280 million. 280 million hemoglobin molecules inside one red blood cell on average. Okay, so you think about it. The structural organization and relationship of size of objects and what can fit inside another object. Hemoglobin is small enough that it can fit 280 million times inside a red blood cell. Now remember, all we're getting here is one dimension, 8 micrometers diameter. We're not giving you length, width, and depth, so we're not talking about the volume of these objects. Okay. We have X chromosomes, right? 7 micrometers long on average. We're getting a little bit larger, a little bit larger. Here's a skin cell. Take a look at your arm. Obviously, it's covered with skin cells. The average skin cell is about 30 micrometers wide, 30 millionths of a meter wide. Okay, we have a sperm cell up here, which is 60 micrometers long from head to tail. We have photoreceptors, which are 100 micrometer long, which are found at the back of our eyeball. Okay, we have rods and cones, which give us black and white vision and color vision. You see a human egg cell here, 130 micrometers wide. Take a look at how gigantic the human egg cell is compared to the sperm cell, okay? Next, we have some other microorganisms, paramecium, 210 micrometers long. The amoeba proteus, 500 micrometers on average as an average diameter. And you say, this is all well and good, but does this have anything to do with the human body? Well, folks, your body is absolutely loaded with microorganisms, which are part of your body's physiology. They influence and alter your body functions. Okay, so here's a question for you in the chat window. How many human cells make up your human body? How many human cells make up your human body? Obviously, the answer for your body is different from my body, which is different from your child's body at the house because they're a lot smaller in size, right? The point is, we're not going to have one single answer that's correct for everybody. But if we were to get a ballpark range of values here, what would we say? How many human cells make up your human body? Okay. Brendan, I like your answer very much. Okay. So the typical range that I've seen is about 50 to 75 trillion. About 50 to 75 trillion human cells make up the typical human body. 50 to 75 trillion human cells in your human body. Now you think about that for a second. I don't know about your experience, but in life, if you get 2 or 3 people in the same room having a conversation about a topic, typically you have 2 or 3 people that don't agree with each other, right? And you think about it. 2 or 3 people can't agree with each other, and we fight and bicker all the time, politics, all that kind of ugly stuff. Your body is made of 50 to 75 trillion cells that actually have to get along. They actually have to agree with each other. They actually have to share a common purpose and common mission, or you don't get to enjoy the life that you do have. So it's kind of amazing what Mother Nature achieves. So getting back to the story of microorganisms, okay? Scientists believe that in your body, you have roughly 10 microorganismal cells or virus particles, because viruses are microorganisms too. You have about 10 microorganismal cells or virus particles for every 1 human cell. So if we have 50 trillion human cells that make up your body, that would mean that you have 500 trillion microorganisms living in your body and on the surface of your body at any moment of time. And those hundreds of trillions of microorganismal cells heavily influence the overall health of your body. Okay, let's continue. We're getting bigger here. So amoeba proteus, we said, is 500 micrometers. Then we get to a grain of salt, something you have a little bit of experience with, 0.5 millimeters. A millimeter is 1,000th of a meter. We're getting a little bit bigger. A sesame seed you might find, on a bagel or something. Grain of rice, 8 millimeters. A coffee bean used to brew your coffee in the morning, 12 by 8 millimeters. And then a typical word processing document, Times Regular 12-point font. Okay, and this is where we stop. Okay, so folks, I don't expect you to memorize all these things. I don't expect that. But what I want you to do is start seriously thinking about the relative size of objects that are in a cell, in a tissue, in an organ, in your body. You need to start appreciating those relationships because, if you were to tell me that an atom is larger than a mitochondrion, that would be heresy because it would be horribly incorrect, right? So you need to think about these things. Okay, so let's document a little bit of what we've talked about here and we'll move forward. So we're going to go from small to large here, okay? So the first level of organization is going to be atoms. And an example of an atom would be carbon atoms, hydrogen atoms, right? Oxygen atoms, et cetera, et cetera. If we take atoms and we link them together with chemical bonds, we build molecules, okay? By molecules, we're going to have things like glucose, we're going to have things like phospholipids. And so let me ask you, give me an example of any molecule you know of. Any molecule you know of. Just tell me the name of a molecule. Okay, so water, carbon dioxide, these are fair games. Okay, let me put a couple more in here. Insulin, right? Insulin's a molecule. tRNA, that was in our web animation a few moments ago. So, you know, just so that you have some examples of what molecules really are. Atoms are the smallest things. You put them together, you build molecules. If we put molecules together, we're going to have something that is not talked about in figure 1.1 of your textbook. Okay, so I'll reference figure 1.1 here, but I'm going to give you a little bit more detail than figure 1.1. So we have atoms, we put them together to build molecules. If we take molecules and we organize molecules into functional entities, then we can build structures that are larger than molecules because they're made of multiple molecules, but they're still smaller than the cell. So they fit into a cell. So here we would have nanostructures and nanomachines. And when I talk about nanostructures and nanomachines, I'm talking about things that we would most appropriately measure with the scale of nanometers, which you might recall is 1/1000th of a meter. So an example of a nanostructure would be, you know, organelles like lysosomes. Okay, organelles like lysosomes. Okay, an example of a nanomachine would be a machine such as a ribosome. Okay, such as a ribosome. These would be examples of nanostructures and nanomachines. So a lysosome is made of many groups of molecules. So they're larger than individual molecules, but they still fit inside a cell. A nanomachine, like a ribosome, is made of many molecules that interact. So they are larger than an individual molecule, but they're still smaller than the overall dimension of a cell, so they fit inside cells. Okay. All right, so that would be number 3 here. I should have put number 3 down. Let's continue. So if we take the nanostructures and nanomachines and organize them into a collective framework that does something meaningful, then yes, we're going to get to the level of organization that we would refer to as cell. So 4 is cells. Examples of cells would include obviously red blood cells. You know, the animation, the video had skin cells, which are epithelial cells. And of course, there were other cells which were microorganismal cells like bacteria, you know, E. coli bacteria, et cetera, et cetera. These would be examples of cells, okay? All right, so, you know, even though bacteria is smaller than a red blood cell, bacteria is still very much larger than a ribosome, which is a nanomachine, which is made of many proteins, which are molecules, which are made of many atoms, which are even tinier things, right? So we'll start talking about the hierarchy of structural organization and the size and relatedness of things. If we take cells and we group them together, we will get to tissues, okay? Tissues are made of many cells working together to serve a common purpose. And so there are many types of tissues, but there's really only 4 major categories of tissues. And we'll list them out here, and you'll start diving down into the details of tissues in chapter 4. They are epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Those are the 4 major categories of tissues. So if we group cells together, we build tissues. Tissues are made of cells. Cells are made of nanostructures and nanomachines. Nanostructures and nanomachines are made of molecules. Molecules are made of atoms. Okay. Excuse me. Number 6 would be organs, right? If you organize tissues together, you form 3-dimensional objects that are organs. Okay, so you can pick your favorite organ. We can talk about the heart, spleen, right? Kidneys, etc., etc. Believe it or not, your skin is a very large organ in terms of mass and surface area. Skin is an organ. Believe it or not, we're going to be, you already know we're diving into the bones of the skeleton. Every one individual bone of your skeleton is an organ. Each bone of your skeleton is an organ. Okay? Bones are organs. We don't think of them that way because we just think, oh, they're bones, they're skeleton, but they are organs. Okay? If we take the organs and we group them together in some organized fashion to accomplish some organized goal, then we would have organ systems. That would be number 7. You will find from reading chapter 1 that there are in fact 11 organ systems. I'm not going to list them all out, but you will be reading about them in chapter 1. Okay, and so just as an example, if you have your digital textbook open and you go to table 1.2, table 1.2, you'll start to see all the organ systems listed out there, right? So we have the integumentary system. And I'm just randomly flipping around here. We have the endocrine system, with hormones. We have the digestive system, okay, which is probably familiar to you, etc., etc., etc. Okay, if we group all of these organ systems together in a way that provides a complete structure or picture for us, then we get to the complete animal body, which for our purposes in our course, the one we're interested in learning about is the human body, right? All right. So there we have it, folks. That's a rundown on the structural organization and relationships of things from the smallest entities of atoms the whole way up to your complete entire human body. Folks, do you have any questions right now? Okay. So, you're currently reading chapter 1. If you haven't already done that, you will be getting some homework in lab that tickles you with some questions on this content. You're going to be starting to really wrestle with this as we get up to speed in the semester. Okay, so you will find that the textbook has a lot of information. And honestly, there's no way that I can describe every piece of information in every chapter, okay? Typically, I'll give 2 lectures for a chapter. So I will pick and choose the topics that I want to talk about. Please don't think that that means other topics aren't important. They are. It's just that I have to choose how to use our time together. So please don't think just because I didn't lecture on it, it's not going to be tested, because that's not true. There will be things that I never talk about that will still be on your exams, because your responsibility as a student is to completely read the chapters and to study the chapters in their entirety. Okay? That being said, you can take a strong hint from the content that I do cover in class that definitely those topics will be strongly tested on exams. Okay? So any questions right now? Okay. So here's my question for you in your textbook. What is the title of section 1.4 in chapter 1? What is the title of it? Awesome. Thanks so much, everyone. I appreciate you following along. It is homeostasis, right? And so this is the second and final major topic of this chapter that I'm going to spend class time on. We won't even get it finished today. We're going to have to finish it next time. But let's begin this conversation. So let's talk about homeostasis. Okay. Okay. So like all good things, perhaps I should begin with a definition. So homeostasis refers to the state of balance, or perhaps a better word would be equilibrium inside your body. That your body temperature is not too warm, it's not too cold, that your blood pH is not too high, it is not too low, that your blood pressure is not too high, it is not too low, that the glucose concentration in your bloodstream is not too high, it is not too low. We're talking about the Goldilocks phenomena, right? Goldilocks went and got the porridge, and we had to be sure that the porridge was not too hot, it was not too cold. Okay, so homeostasis refers to the state of balance or equilibrium, if you prefer, that exists inside the body for different parameters, such as blood pH, glucose concentration, temperature, etc., that is maintained by regulatory machinery that exists inside cells, tissues, organs, and organ systems. Okay, so the idea here is that there is regulatory machinery that exists in the organ systems to maintain homeostasis on, let's say, body temperature. Well, obviously, the organ systems are made of organs, so that machinery is part of the organs. Obviously, the organs are made of tissues, so that machinery is part of the tissues. Obviously, tissues are made of cells, so that machinery at the molecular level is part of the anatomy of cells. Okay, so homeostasis refers to the state of equilibrium that exists inside the body for different parameters that we could discuss. And this homeostatic condition is maintained by regulatory machinery that exists in our body's anatomy. Okay, so let me give you a slightly more precise example of homeostasis. Let's take a look at blood blood sugar. Okay, so hold on a sec. Okay, so I'm going to ask a question in the chat window. Here it comes. What is the homeostatically normal range of resting blood sugar? So what I mean by that is when you went to bed last night and slept for 7 or 8 hours, hopefully, and you wake up in the morning, before you eat anything, before you have any breakfast, what should the normal range of blood sugar be? Okay, so I see Brendan mentioned 85, and I'll hold on commenting there. Anybody else? Okay. All right. So notice, okay, good. We're getting some good values, and I appreciate that. And notice the question, notice how the wording of the question was given. What is the homeostatically normal range of resting blood sugar? So can you answer a question about range with an individual value? Okay, not really, right? You need to give me a range. You need to give me at least 2 numbers with some distance in between those 2 numbers, okay? But all of you did state numbers that are within the appropriate range, and I appreciate that, okay? And so the range of resting blood sugar, you're going to get different answers on what is normal and healthy. And so I don't want to get into a disagreement between experts, but we'll stay with what your textbook tells us. Your textbook tells us that the resting range of blood sugar is 70 to 110. Now I'm going to keep picking on you folks. Okay, I'm going to keep picking on you because most of you in your answers ignored the units. Without the units, the number means nothing, right? So Ashasia, I see that you applied a unit of milligram. That's good, but it's only half correct. Jared, you gave me milligrams per deciliter, and that's a better answer, but there's still something wrong with it. Okay. Okay, there's a huge difference between deca and deci. Yep, absolutely, Jared. Okay, so let's type this down. Resting blood sugar is homeostatically normal in the range of 70 to 110 milligrams per deciliter of blood plasma. Okay, resting blood sugar is homeostatically normal in the range of 70 to 110 milligrams per deciliter of blood plasma. Okay, so I want to make you aware, be aware of the following. Okay, in your textbook, I want you to locate Appendix C. Locate Appendix C in your textbook. Locate Appendix C. And I'm going to ask a question. As you locate Appendix C, I'm going to ask you a question to see if you're accessing the content correctly. Okay. What is the US reference value? What is the US reference value for creatine in females? What is the US reference value for creatine in females? Okay. All right, Brendan, you got me the number, but you didn't get me the units. The units matter. The units matter. The units matter. Okay. Okay. Thank you very much. Okay. Good, 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 good. Awesome. Does everybody see that it is in fact 0.35 to 0.9 milligrams per deciliter? Okay. Now, of course, you might say, I don't even know what creatine is. Well, don't worry about it. Right now, you don't have to know what it is. Okay. But the point that I'm making right now is, can you access Appendix C and can you read it correctly? Okay. And it seems like many of you do have done that. And thank you for participating. So, folks, you don't have to memorize these things. You simply have to be able to look them up, right? You don't have to memorize them. You have to look them up. The whole reason we have appendices in textbooks, the whole reason we have reference texts and tables and charts and databases and electronic resources like Google and things like that is so we don't have to memorize it, right? We can look it up. And so that's what I'm pointing out. Be aware of Appendix C. And if I ask you something off the wall about a US reference value, you'll be saying, oh, I know what he wants me to do. He wants me to go look it up in Appendix C, right? That's what he wants me to do. Okay. Any questions about locating Appendix C? Be aware of Appendix C in the textbook. Okay. So we're talking about homeostasis and the idea that certain body parameters like blood glucose or temperature, or any number of other things we could talk about, needs to be within a certain range to be considered healthy. If the value of that parameter falls outside that range, then you might not be healthy. You might actually feel sick. And in fact, when you go to the hospital and you're sick, don't they often do a lot of blood work? Don't they do a lot of blood work to measure certain things to see what certain parameters are, like, what your level of liver enzymes might be in your blood, something like that. Okay. The whole idea is by measuring things, we can tell potentially what is wrong with your body and how the medical staff need to support your body to heal and get better. So homeostasis is a very important topic. And we need to talk about this. So the control of homeostasis takes place through feedback systems. Okay, and so I'm going to define a feedback system for you. Okay, so feedback systems, when you say the phrase feedback system, you are referring to all the components of the body. This is a system made of multiple components. And when I say multiple components, I'm talking about cells, tissues, organs, organ systems. Okay, so I'll be verbose right now. Cells, tissues, organs, organ systems. This is a system made of multiple components that work as a team. So they're working together. This is teamwork. This is a collective interaction to monitor a body condition. By body condition, I'm talking about a body parameter, such as blood glucose levels, body temperature, liver enzyme values, carbon dioxide concentration in the blood. Okay. Okay. So all of these components work together as a team to monitor the condition. And then if that condition is determined to be out of the homeostatically healthy range, that anatomy will take corrective action. So a feedback system is made of components of the body that work as a team to monitor body conditions and which take corrective action to adjust the body condition if it is outside homeostatically healthy values. Okay, so that's kind of a long-winded definition. Let me read it one more time. A feedback system is the system made of multiple components, multiple pieces of your anatomy, cells, tissues, organs, organ systems that all work together as a team to monitor the status of a particular parameter of your body, such as body temperature. That parameter will be monitored, and if it falls outside a normal homeostatic range, which is established by the physiology of your body, then that anatomy will take corrective action to restore you back to health. If your body cannot restore itself back to health, then you're going to get sick and you might need some medical care. You might need medical intervention. Okay, so as I continue in chapter 1, around figure 1.2, you're going to see some text under a heading called feedback system, and it's going to tell you what the basic components of the feedback system are. These components are also outlined in figure 1.3, and we'll talk more about these things as we continue. But let me tell you about the actual components of a feedback system. A feedback system is typically made of 3 major components. And so the first would be a receptor. Okay, so a receptor is going to be a body structure. That body structure typically includes a nerve ending. So there's going to be a neuron involved, right? Because the neuron has to transmit information about the body condition being monitored to the major control center of the body, which is your brain. So a receptor is a body structure which almost always contains a neuron that monitors changes in a body condition. And again, that condition could be carbon dioxide levels in the blood, pH levels in the blood, liver enzymes in the blood, a whole bunch of different things, blood pressure, okay? That monitors changes in a body condition and communicates that information to a control center, okay? Now, the control center is often the brain, but in some contexts, it can be other organs, okay? So I'll just put in here, often the brain. And there's a bit of terminology here you need to be aware of. When the receptor, okay, communicates information to the control center, when you deliver information into the control center, the name of that communication is given a special phrase. It's called afferent communication. So communication that moves information from the receptor into the control center is known as afferent communication. Okay, afferent communication. All right. So let me give you just a very brief example here of something that might make some sense in terms of what a receptor actually is. Okay, so an example of a receptor could be the taste buds on your tongue. The taste buds on your tongue are receptors for your food molecules, right? Taste buds on the tongue are receptors. They're going to detect the food molecules you're placing into your mouth when you enjoy a meal. Now, you may not know what taste buds are actually made of from an anatomical perspective. And I promise you, you will by the end of this course. But suffice it to say that taste buds are actually made of groups of cells that contain a neuron. And so when the neuron is stimulated by the food molecules, it generates an electrical current that flows up the spinal cord and into the brain. And your brain processes that information and says, oh, you've just put something sweet into your mouth. Maybe you're eating a sticky bun or something like that, okay? So the taste bud would be an example of a piece of anatomy, which is the receptor. The taste bud is a piece of anatomy. It's part of your tongue that is the receptor. What is it monitoring? It's monitoring the molecules of food that you're putting in your mouth, okay? All right, so taste buds would be an example of a receptor. Any questions so far? Okay, let's keep going here. We're getting close on our time. Let's talk about the control center. Okay, the control center is a body structure. Again, it's often the brain, but it's not always the brain. It's often the brain, but not always the brain. This is a body structure, often the brain, that receives the information from the receptor and processes that information to determine if the body condition or parameter being measured at that moment in time is within normal homeostatically healthy ranges. Now, of course, your brain or your blood or your pancreas, whatever we're talking about, it doesn't use the terminology that we humans use, right? It just thinks in terms of biochemistry, biophysics, thermodynamics. It's not going to say, oh, okay, the blood glucose level is 80 milligrams per deciliter, so that's okay. It's not going to, your brain is not going to think about 80 milligrams per deciliter. It's just going to think about the chemistry and physics of whether that concentration of sugar is a good concentration of sugar to have. Okay, so the control center, again, often the brain, this is a body structure that receives information from the receptor, processes the information, and communicates to an effector how to respond to that information. Okay, the response that is given, so let's put it this way, the instructions from the control center can be to take no action, which means everything is homeostatically normal, or take corrective action, which means something is wrong. A body condition or parameter has fallen outside a healthy range. Okay, so the instructions from the control center can be to do, one of two things. Take no action or take corrective action, okay? Now, just like there was special language of afferent communication moving information from the receptor to the control center, when the control center communicates information away from itself to what will be called an effector, that information, that flow of information is known as efferent communication. Okay. Communication away from the control center toward an effector is known as efferent communication. Okay, so folks, I'm looking at my clock here and I see that I'm a minute past time, so I need to stop. So please let me just tell you what we're going to do here moving forward. Okay, what you can expect on next Tuesday is that I will continue on the topic of homeostasis, probably for 30 to 40 minutes, because this is really an important idea of understanding how the machinery of your body monitors body conditions and adjusts them if it's necessary. So we'll spend 30 to 40 more minutes on homeostasis to field any questions you might have. And if there's no questions, I will start chapter 2 on Tuesday. Okay, so please be reading into chapter 2 over the weekend. If you can't get it finished, please just get a few, try to get 10 pages of it read or something like that. Cassandra, absolutely yes. I see your question, no problem. Do you have any questions at this time before we sign off? Okay, folks, have a wonderful, safe weekend, and I will see you next week, okay? Bye for now.